And I'm going to do sort of a few minutes each, first of all on cell sorting, and then I'm going to talk about some very recent work on tumor distributions in breast cancer. So let's start with the cell sorting and call out to Saraswat, who was just finished his PhD and now starting a postdoc in Dresden, who did a brilliant job on this project and actually with other collaborators on the next one as well. This is the experimental system we were guided by. If you have two different sorts of cells, in this case, purple cells and green cells, then they tend to, at least they tend to order, not completely, but you tend to get microphase separation. The difference between those cells is one of them are wild type cells and the other one has been knocked out. The E cadherin at the cell-cell junctions has been partially knocked out. You get this happening in cell spheroids as well. These are cancerous cells and normal cells mixed together, and you can see that you definitely get sorting of the two different sorts of cells. So we wanted to ask, can this sorting be due to activity? The official story, the official sort of story that's in the biological literature is that it's very like passive phase separation, that the cells have slightly different surface tensions between each other, they have different interactions, if you like, and so they tend to phase separate just like an oil-water mixture. But these things are active. Can activity give you sorting? And we know that activity actually can give you sorting because many of you will have heard of MIPS, when particles tend to form dense clusters if they're active. And people will also have thought about some very interesting work by Owen Frey, where he just mixed together two gases, I mean, in a computer simulation, mixed two gases which were maintained at different temperatures, and these things sorted. So it's a bit like saying two systems with different activities will sort. Does that work if I have a mixture of active nematics? So we had to extend the theories of liquid crystals, of active liquid crystals, to a two-fluid model so that I've got two different fluids which can actually sort themselves out. Each component has to be compressible. It has to be able to have a different concentration in different places. Whereas the whole thing, the center of mass fluid, can just be a normal incompressible fluid. And the two fluids have to have relative flows, which allow the concentration field to change. The A and B components, if you like, active and passive components have to move relative to each other. And then we also put a viscous drag between the components just to keep things reasonably, the relative flows relatively small. So what happens? If I mix an active and a passive fluid, there is absolutely no thermodynamic anchoring, which is trying to order them. They're in a potential well, which has a minimum, which says it wants to be 50-50 everywhere. What happens? And the answer is that this happens. The yellow bits are active rich regions, and the blue bits are active poor regions. Here's the concentration difference, the average concentration difference as a function of time. This is when it gets going. And then you can see that there is a well-defined concentration difference. It's pretty small, though. It's about 6%. So what is happening, there's some sort of process which is causing these things to phase separate, active phase separation, but then they're also doing active turbulence, and that's mixing them up again. Just to sort of check that works is we looked at the amount of separation as a function of activity, and you have more activity, they get more separated. If instead of mixing an active and a passive system, we mixed an active extensile and an active contractile system, then again you get this separation, but it's much, much stronger. Essentially, it's all extensile here and all contractile there. And if we put on friction, that sort of kills the active turbulence a bit, and you get larger and larger domains. So you can get separation just due to activity. So the next slide tries to explain why. And this is based on this active, it's an instability, and it's an instability which relies on flows, and it relies on active anchoring. So here's my system. Let's consider a small fluctuation so that this side is a bit more active and that side is a bit more passive, i.e. there are more active molecules, grey ones, on this side than there are on that side, or colloids, more active particles on this side than on that side. This system is extensile, and this is an interface, and so you get active anchoring. And from an extensile system, active anchoring means these things line up parallel to the interface. But they're extensile. And for extensile particles, we know the flows go out from the ends, I haven't put that on, and in from the sides, which is these red arrows here. And you can see that the net flow is in that direction. So more active material is going to flow in this direction, and you're going to get this instability so that the active side becomes even more active and so on. And you're going to get this instability forming until the active turbulence mucks it up. Same thing actually happens for a contractile system because you have two changes of sign. The active anchoring lines things up perpendicular to the interface, and then the flows are into the ends, and those two changes of sign means you can use the same argument. You also get a sort of active wetting type thing. These things not only sort in the middle here, this is now a box, in the middle here of the box, you get this active turbulence like we had before. But if you look at the edges, here they tend to be yellow, and here they tend to be blue. So you're getting this active sorting to a boundary. If it's planar anchoring, if it's an extensile fluid with planar anchoring, it likes going to the boundary. And with homeotropic anchoring, it likes going away from the boundary. And that's because, again, because of a gradient. Now the gradient is that the pneumatic order decreases away from the boundary because it's imposed at the boundary, either imposed thermodynamically or imposed by active anchoring, doesn't matter. And so because it decreases away from the boundary, that gives you an active force towards the boundary. Let me just show you my favorite picture that one day I want to explain. This is a starfish embryo, which has basically been put in a blender. And the two sorts of cells in the starfish embryo have been all mixed up. And if you just leave them there, they reassemble and they microphase separate. And then you get this sort of sorting to a boundary thing. And you end up with a system which looks back like a starfish. And it's pretty amazing that nature can do that. And maybe we're trying to understand better what's going on there. So that was the first story. Now I'll tell you something we've been thinking about lately, which is very different, a bit different from anything we've thought about before. So it's been fun, and that's looking at tumor distributions in breast cancer. And again, Sarah Swat was responsible for the simulations and explaining the biology to us and doing all the analysis of the data that I'll show you is Pablo Gottheil in Joseph Kusser's group in Leipzig. So what happens in breast cancer is one starts, you start with a tumor like this, and it's a well-defined tumor, and around the tumor is extracellular matrix polymers, which basically imprison the tumor. And at this stage, really, it's not very dangerous. Then with the progression of time, the tumor breaks up in a way that we don't understand. Presumably it just sort of explodes or the cells suddenly start to escape. And then you end up with a situation like this, where the cancerous tumors are much more diverse. You get these smaller tumors and you get a lot of them, and they get very tortuous shapes. So these dark purple things are cancerous cells, and the pink background is extracellular matrix. And if you sort of zoom in on one of these clusters, what you find is a cluster of cancer cells in purple here, which have a velocity and which presumably are acting as an active pneumatic because they're cells. And surrounding that is the extracellular matrix, which again tends to have an ordering to it. And so it's a rather disordered pneumatic, but it's still pneumatic. It's a load of filaments, and so it's a pneumatic. So our question was, what do the distribution of these invasive breast cancer lesions look like? And the data we had were histological images from many patients, which were cross-sections across the malignant tissue, two-dimensional cross-sections, 10 to the 5th different clusters. So the data is incredible. It doesn't have error bars. The data looks like this. And what Pablo did is look at these clusters and find their sizes and shapes. But the problem with this is those images are static, because these things move very, very slowly, and you can't expect a patient to sit there for a year or so while they move around. And so the distribution of sizes is known, but the dynamics is not known from this data. So we wanted to see if we could think of a model, or not think of a model, try and write down what we felt was a sensible model, and see if it can give the same sort of distribution as there is here. So we modeled the cancer cells as an extensile active pneumatic, assumption, but maybe a reasonable one. The extracellular matrix is modeled as a viscoelastic pneumatic, a thing called the Kelvin-Voigt model. That's often used for extracellular matrix. And because we're doing dynamics, we're solving a coupled set of differential equations. We can get dynamical information. And so if we look at our dynamical pictures and sort of take snapshots, can we compare the sort of clusters we get, which is a picture here, to what's seen in the breast cancer? So I'll show you some simulations. This is, we start from a completely random configuration, and then we end up with a steady state where we get a distribution of clusters. If we start with a pneumatic blob, just to see if we end up in the same place, we end up in a very similar, in fact, as far as we can tell, the same final steady state. And our data is from the final steady state. This is time evolution in those simulations. What's happening is that these little clusters are forming. These are the active clusters. They're effectively doing active turbulence. They're either coalescing. These red arrows show ones which are coalescing in time, so they're getting bigger. This black arrow shows ones which are splitting up in time, so that one's getting smaller. And at least visually, we can compare what's happening between the simulations and the breast cancer clusters. So this is medical data. These are our simulations. Of course, we don't know the activity here, so we're guessing that intermediate activity matches that. We're guessing with high activity, seems to give us smaller clusters, matches that. So can we get better data? And this is my essentially last slide. this is the sizes that we get. This is the cancer cluster size, and this is the probability of different sizes. The red is from the histological data, from this medical data. You can see that really there are no error bars here because there's just such a huge amount of data. The simulations are the blue data here. Again, we get a power law decay of the cancer cluster size. Goes wrong there. That's because we can't resolve the smaller clusters in the simulations. Power law doesn't actually mean that much. We had to average over activities, and if you average over activities, you almost have to get a power law. This, I think, is more convincing, where we look at the shape index of the clusters. So the shape index is small here, it's not very tortuous, up to a shape index here where they're very tortuous. And here we can get essentially an exact map between the red histological data and the blue simulations. And this correlation between size and shape, small ones are round, big ones are much more jagged, is seen both in the data and in the simulations. So perhaps, I mean, it's just a new way of looking at these pictures, which are the way medics judge how bad breast cancer is. It's a way which says that what we're seeing here is because of active pattern formation. That's a bit surprising, because certainly my first thought is, why haven't you put growth in these simulations? Why isn't growth important? But if you go back and look at the figures, growth seems to be too slow. It's difficult, the numbers are not accurate, but the growth seems to be too slow to talk about what's happening here. So hopefully this will be the beginning of seeing if these ideas can work in model systems and in other forms of cancer. So I'll stop there, put up some references in case anyone's interested, and thank you very much for listening, and happy to take questions.